Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about mass spectrometry. Mass spectrometry. So, it is an analytical chemistry technique that helps to identify the amount and type of chemical present in your sample by measuring its mass to charge ratio and abundance of gas phase ions. In this instrumental technique, first sample is converted to rapidly moving positive ion by electron bombardment and charged particles then separated according to their masses. A mass spectrum is a plot of relative abundance against ratio of mass and charge. These spectra are used to determine the elemental and isotopic signature of the sample, the masses of particles and of molecules to elucidate the chemical structure of molecule and other chemical compounds. So basically at first the sample are converted into charged particles, then separated on the basis of their mass. Then what is the principle? In this technique, molecules are bombarded with beam of electron at first. Molecules are ionized. They are converted into ions and broken off into many fragments, some of which are positive ions. Each kind of ions has particular ratio of their mass to their charge. For most ions, the charge is 1. Thus, mass to charge ratio is simply the molecular mass because charge is 1. So, the ion pass through magnetic field and electric field to reach the detector, then where they detected and signals are recorded to give mass spectra. Okay. So it is the graphical representation of mass spectrometry. At first samples are injected, then vaporized, then bombarded with electrons, then accelerated ions. These are accelerated ions. Then these are magnetic field applied to the ions. Then detector detect those ions. Okay. So what are the principle working, how it works? In a typical procedure, at first simple, maybe solid, liquid or gas, at first ionized by bombarding it with electrons. This may cause some of the sample molecule to break into charged fragments. These ions are then separated according to their mass to charge ratio, typically by accelerating them and subjecting them into electric or magnetic field. Ions of same mass to charge ratio will undergo same amount of deflection. The ions are detected by a mechanism capable of detecting charged particle. Results are displayed as spectra, as graph of the relative abundance of detected ions as function of mass to charge ratio. Atoms or molecule in sample can be identified by correlating known masses to identified masses or through characteristic fragmentation pattern. So this is the graphical representation of the whole process of mass spectrometry. At first samples are introduced, then these are the sample are converted into gas phase ion, then by analyzer the samples are analyzed, then detector detect the ions present in that sample. Then this is the data output in the form of graph. Okay. So what are the applications? Environmental monitoring and analysis in the field of geochemistry, in the field of chemical and petrochemical industry. Identify the structure of biomolecules such as carbohydrate, nucleic acid, sequence biopolymers like protein and oligosaccharides. Then determine the molecular mass of peptides and proteins and oligonucleotides. Monitoring gases in patient's breath during surgery. identification of drug abuse and metabolites of drug abuse in blood, urine, and saliva, analysis of aerosol particle, determination of pesticides residue in food. So these are the applications of mass spectrometry. Okay, so it is all about your mass spectrometry. I hope you liked it. If you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's tutorial. Thank you.